The complete oxidation of fatty acids to CO2 and H2O takes place in 3 stages. What are they? Where do they take place? One, oxidation of longer-chain fatty acids into 2-carbon fragments in the form of acetyl-CoA. B. Oxidation in mitochondrial matrix. 2. Acetyl-CoA CO2, the Kakin matrix. 3. Transfer of electrons to the mito-respiratory electron chain, OXPHOS in inner mitochondrial membrane. What is the function of the carnitine shuttle? What is added to fatty acids to begin this process? Do fatty acids with 12 or less carbons need the shuttle? The carnitine serves as transporter so fatty acids with more than 12 carbons can get through the membrane. CoA is first added to the fatty acid to create fatty acyl-CoA. What does carnitine acyltransferase 1 do? What is it inhibited by? Carnitine ACT transferase 2 transfers the acyl group that was attached to fatty acid back to CoA. Acetyltransferase 2 is inhibited by malonyl-CoA, the first intermediate in fatty acid synthesis, prevents the simultaneous synthesis and degradation of fatty acids. What is the importance of keeping pools of CoA separate? By keeping pools of CoA separate, there can be different concentrations. Cytosolic CoA can be used for synthesis and mitochondrial CoA can be used for degradation. Briefly describe 4 steps of beta-oxidation. Step 1. Dehydrogenation by fat via dehydrogenase, oxidation of the fatty acyl-CoA and formation of double bond. Step 2. Hydration via hydrase where water adds across the double bond. 3. Dehydrogenation by NAD via dehydrogenase converts hydroxyl group into a keto group and NADH is formed. 4. Dialysis via acetyltransferase thiolase B ketoacyl-CoA cleaved by thiol group of another CoA, inserted between C2 and C3 to make acetyl-CoA. What type of double bond is formed in step 1 of beta-oxidation? What happens to the FADH2 formed? What happened to the NADH formed? A trans double bond is formed in step 1. The FADH2 formed feeds into the electron-transferring Flav protein, ETF. The NADH goes to the electron transport chain. For each round of beta-oxidation, how many FADH2 and NADH are formed? How many acetyl-CoA are formed? One FADH2 and one NADH and one acetyl-CoA, 2 on the last cycle. For each round, for each acetyl-CoA made that goes into the CAC, you get 3 NADH, FADH2, and 1 GTP, ATP. For a fatty acid with 18C and no double bonds, how many acetyl-CoA are formed? FADH2, NADH. If these products are completely oxidized, how many ATP will you get? Eight NADH, 8 FADH2, 9 acetyl-CoA, will get a total of 120 ATP. 1 ATP each will get a total of 120 ATP. 2 used for carnation shuttle. How does beta-oxidation lead to the formation of metabolic water? Water is formed through the process of electron transport and oxidative phosphorylation. Why do monounsaturated fats pose a problem in beta-oxidation? How is this overcome? Unsaturated fatty acids have CI's bond, which prevent the formation of a trans double bond in step 1. This is overcome by using an isomerase to convert it to a trans double bond. In a polyunsaturated fat with two adjacent double bonds, in addition to isomerase, what other type of enzyme do you need? In polyunsaturated fatty acids, a reductase is also needed to create a new single double bond, but because it's in the wrong conformation, another isomerase will also be needed to convert it to trans. What problem is posed by odd-numbered fatty acids? In addition to acetyl-CoA, what else is formed? What is the fate of this 3-carbon molecule? Odd-numbered fatty acids will oxidize the same as even-numbered ones until the end of the chain, when they become acetyl-CoA and propionyl-CoA. Propionyl-CoA can be acted upon by B-oxidation by being converted to succinyl-CoA, which can then enter the CAC. The carnitine shuttle is rate-limiting in the oxidation of fatty acids. What is malonyl-CoA and how does it regulate entry of fatty acids into the mitochondria via the carnitine shuttle? Malonyl-CoA is the first intermediate of fatty acid synthesis from acetyl-CoA in the cytoplasm. Its levels increase when our carbohydrate levels increase. Excess glucose made into Fa. Malonyl-CoA inhibits carnitine acetyltransferase 2 from shuttling Fa into the mitochondria and being degraded. What is ACC? Briefly describe how glucose regulates its activity. ACC is the first enzyme in the synthesis of fatty acids, so it makes malonyl-CoA. High glucose will lead to high insulin and unphosphorylated ACC, which will be activated. Low glucose will lead to high glucagon levels and phosphorylated ACC, which will be inactivated and unable to make malonyl-CoA, meaning the carnitine shuttle will be activated and fatty acetyl-CoA can go into the mitochondria. What are ketone bodies? The three major ketone bodies. Ketone bodies are fuel that are synthesized in the liver during fatty acid oxidation. The three major ketone bodes are hydroxybutyrate, acidoacetate, acetone. When is acetyl-CoA converted into ketone bodies? Where are they produced? What is their function? Acetyl-CoA is converted into ketone bodies when CAC intermediates are depleted. They are produced in the liver. Their function is to be converted to acetyl-CoA and used in the CAC. What is ketosis? Ketosis occurs when acidic ketone bodies cause too much lowering of blood pH. Starvation causes the overproduction of ketone bodies. Briefly describe how in each case. In starvation, gluconeogenesis occurs to make up for the lack of carbohydrates, which in turn depletes CAC intermediates. Acetyl-CoA is diverted to produce ketone bodies when CAC intermediates are low. Also, growth hormones secreted to lipases in adipose tissue to provide fatty acids for oxidation, which produces even more acetyl-CoA. Diabetes causes the overproduction of ketone bodies. Briefly describe how in each case. When insulin levels are low, ACC is inactive because it becomes phosphorylated. This causes malonyl CoA levels to fall and beta-oxidation to occur, so acetyl-CoA levels increase, leading to production of ketone bodies. Because insulin is low, cells can't take in glucose. As glucose levels fall, gluconeogenesis is used, which depletes the CAC intermediates and leads to more formation of ketone bodies. What Greek letter is designated to the last methyl carbon of a fatty acid chain? Omega. Fatty acids form blank in water. Micelles. Fatty acids are stored as energy reserves through an ester linkage to glycerol to form blank. Triacylglycerols. True or false? Triacylglycerols form neither micelles nor bilayers, are not amphipathic, and do not have polar hydrophilic and hydrophobic groups. True. Blank fatty acids are straight while blank are kinked. Saturated, unsaturated. What three components make up a phospholipid? Diacylglycerol, phosphate, alcohol. What kind of structure is favored for phospholipids and glycolipids? Bilayer structure. Blank is a steroid, a lipid essential in the plasma membrane. Cholesterol. In humans, the majority of total energy and fuel reserves comes from blank. Stored lipid. What are two reasons for the greater energy density of fats? Hydrocarbons in fats are more reduced and they are not water-bound, as are proteins and carbohydrates. Underscore 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 made by liver and secreted from gallbladder, promotes solvation of triglycerides for hydrolysis, bile salts, pancreatic lipase. True or false? Bile salts are able to promote micelle formation because they are amphipathic. True. Within mucosal cells, triacylglycerols are reformed and combined with protein, producing lipoprotein transport particles called chylomicrons. Chylomicrons in the blood bind to adipose and muscle cells. Membrane-bound lipases. Utilization of triacylglycerols from adipose tissue requires, what, 3 stages of processing? 1. Hydrolysis by camp-regulated lipases. Free fatty acids enter the blood and are bound to underscore 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 for transport to other tissues for oxidation. Serum albumin. Free fatty acids enter the mitochondrial membrane as blank carried by blank. Acyl-CoA, carnitine. Where does beta-oxidation occur? In the mitochondrial matrix. What are the 4 steps in beta-oxidation repeated for every 2 carbons in the fatty acid chain? Oxidation, hydration, oxidation, dialysis. What are the main products of the beta-oxidation cycles? Acetyl-CoA, FADH2, NADH. In oxidation of unsaturated fatty acids, double bonds at odd-numbered carbons are blank. Isomerized. Polyunsaturated fatty acids with a double bond at blank-numbered carbons are reduced. Even. Odd-chain fatty acids yield blank in the final dialysis step. Propionyl-CoA. Ketone bodies are formed due to the accumulation of acetyl-CoA not processed by the citric acid cycle because underscore 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 is used. Oxaloacetate. Blank is the major source of ketone bodies. The liver. The body generally prefers glucose as an energy source, but blank prefer acetate. Heart muscle, renal cortex.